everyone. My name is Danielle Gatti Palumbo, and I'm a clinical pharmacist at Northwell Health Division of Rheumatology in Long Island, New York. And today I'll be presenting on a journal club of an article published in March of 2025 titled Guselkumab Binding to CD64 Positive Interleukin-23 Producing Myeloid Cells and How It Enhances the Potency of Neutralizing IL-23 Signaling. So a little background is that IL-23 is a key cytokine in inflammatory diseases such as psoriasis, psoriatic arthritis, IBD, and also Crohn's disease. And it is produced primarily by myeloid cells expressing CD64 or Fc gamma receptors on these inflamed tissues. And through a transcriptive analysis of psoriasis and IBD, they have found that there is an increase in co-expression of CD64 and also IL-23 in these affected tissues, highlighting that potentially targeting the CD64 on the myeloid cells as a major source of IL-23. And IL-23 works by binding to its IL-23 receptor, thereby then activating its JAK-STAT pathway and then causing an inflammatory response with increase of, your inflammatory cytokines. Now, we currently have two agents on the market, Guselkumab and risankizumab, which are both IL-23 inhibitors. They are both mAbs that target the IL-23 P19 subunit on IL-23. And when it binds to that, it then neutralizes it by preventing it from binding to its receptor. And Guselkumab actually has a unique mechanism of action because it has two mechanism actions. The first one is that it binds to IL-23. So here's my Guselkumab, and here is your IL-23 with the P19 subunit. So Guselkumab first binds to the P19 subunit on IL-23, preventing it from attaching to the receptor and then activating the JAK-STAT pathway. The second one is that it binds to CD64 on the myeloid cells or monocytes. And then also it can capture IL-23 even while being attached to the CD64 on the myeloid cells. However, both of these agents differ in their Fc domain, and that is specifically attributing to the binding of the Fc gamma receptor. That is the receptor that's on the surface of immune cells, and that binds the Fc portion of the IgG antibodies. Guselkumab has a native wild Fc domain, while risankizumab has an engineered mutation Fc domain with LA mutations, and thereby has a reduced Fc receptor binding. So with that knowledge, the study investigators wanted to see how Guselkumab Fc domain interacts with CD64 on IL-23 producing myeloid cells and how it affects its potency in neutralizing IL-23 signaling. In this study, they used a few methods. They used comparative analysis of the Guselkumabs with its native Fc domain and risankizumabs with its engineered Fc domain with reduced Fc gamma receptor binding. They also used flow cytometry, live cell imaging, and also co-culture assays with IL-23 producing CD64, THP1 cells, as well as IL-23 responsive reporter cells. So mimicking a more of an inflammatory state, right? So some key findings that they found was that CD64 and also IL-23 transcriptions are both elevated in inflamed tissues and are actually co-expressed by myeloid cells in psoriasis and IBD. Another finding is that majority of Fc gamma receptors are low affinity. However, the specific Fc gamma receptor CD64 is the only one that has a high affinity for IgG on immune cells, and that CD64 is a primary source of IL-23. So in the study, they used in-flow cytometry, and they found that Guselkumab binds to CD64 on interferon gamma primed or activated monocytes via the Fc domain, while risankizumab did not. And to make sure that this was due to the Fc domain, they actually genetically engineered both of these mAbs and swapped the Fc domains so that they found that the one that had the LA mutation Fc domain still had diminished binding to the Fc gamma receptor, thereby clearly attributing to the Fc domain specifically. What they also found was that Guselkumab had the strongest binding to CD64 on IL-23 producing monocytes and was able to capture secreted exogenous IL-23 even while being anchored and bound to this CD64, which I showed in my little picture before. And then from there, it's able to internalize it and then into its endosomal compartments. And that in the study, they saw in live cell, or were able to see in live cell imaging. However, risankizumab did not show this. Guselkumab, they also found demonstrated enhanced functional potency compared to risankizumab in co-culture of IL-23 producing CD64 myeloid cells and IL-23 responsive cells. So there again is that co-culture setup. So while both antibodies similarly inhibit IL-23 signaling in the standard assays with exogenous IL-23, in the co-culture assay setup, Guselkumab showed that FC-dependent enhanced potency, actually 10 times more potent than risankizumab. And this they attributed specifically to the FC domain. Guselkumab also binding to the CD64, they found did not produce an inflammatory response or release of cytokines, which they thought could happen potentially because of the binding of CD64 could cause like cross-link receptor activation and cytokine production, but it didn't find that in this study. So that was actually pretty good. Some strengths of this study is that they did use multiple complementary approaches. So they used the transcriptomics, they used flow cytometry, they used live cell imaging and functional assays. They also compared two IL-23 inhibitors and mAbs with two different FC domains and then engineered them to swap the FC domain that show that it was clearly contributed solely to the FC domain. So that was actually pretty cool. And then they also applied this to relevant inflamed tissue, using that co-culture setup with the IL-23 and such. Some limitations of this study is that it's mostly in vitro, right? So we would like to see this, further down the line in more of a clinical setting. And then also more direct comparator studies of other IL-23 inhibitors in patient populations that have, increase in IL-23 or CD64 positive cells. And then what is the long-term impact of this? So when we're targeting FC-mediated targeting therapies, what does that have to say about safety and then also immunogenicity in the long run? And then from this study, basically we conclude that beyond simply just looking at the IL-23 inhibition, the FC domain specifically of Guselkumab allowed it to bind to CD64 on IL-23 producing myeloid cells, enhancing its capture of secreted IL-23 and then also internalizing it, thereby enhancing its antibody potency. And this basically supports its use in diseases such as psoriasis and IBD that have a high burden of IL-23 producing CD64 myeloid cells, and that we're seeing recently more in clinical guidelines and also clinical trials that are showing this positive, response with Guselkumab. And also, while not proving in vivo, this basically supports the hypothesis of how FC interactions with the CD64 can contribute to its efficacy and also therapeutic antibodies in inflammatory diseases. I hope you enjoyed that quick summary of this article. And for more journal clubs and podcasts, visit our RAP.org website for more information. Thank you.